morning, everyone. My name is Heather Hargett, and it is my pleasure to moderate the first session, The Science of Infectious Diseases. We'll kick off our session with our first speaker, Dr. Eong. He is the Deputy Director of the Program in Emerging Infectious Diseases at Duke-NUS Medical School in Singapore. He will be speaking to us today about what makes us sick, the virus, or our response to infection. Dr. Eong, take it away.
Hello everyone, and thank you for this opportunity to share some of our work with you. My name is Eong, Prof Ooi. I'm a professor and deputy director at the Program in Emerging Infectious Diseases at Duke-NUS, and I also co-direct the Viral Research and Experimental Medicine Center at SingHealth Duke-NUS Academic Medical Center. And today I'm going to basically talk about our work on trying to understand what makes people sick when they're infected with a virus. Now, take you through some, starting from COVID and all the way to some of the experimental work that we're doing. Starting with COVID, I mean, that probably needs no introduction. Every one of us, of course, know about this disease and the virus that emerged end of last year, or called SARS coronavirus 2 or SARS-CoV-2 for short, starting first in China and then now expanded all throughout the world. In Singapore, we saw our first few cases in late January. In this slide, I'm showing you the first 3 cases that were seen in the Singapore General Hospital. Case 1, incidentally, was the very first case in Singapore. And what you can see from this chest X-ray is case 1 had bilateral pneumonia, whereas case 2 and 3 had no pneumonia. They were completely, they had a bit of symptoms, but the chest X-ray was completely clear. And important to note was that they were all infected with the same virus. In fact, case 1 and 2 probably shared the identical virus, yet one got pneumonia and the other person did not. So then what makes one person get more severe disease when infected with the same virus compared to the next person? So we started to try and understand that very early on in the course of a COVID outbreak. And this is the first paper that was published in our group, from our group, led by Eong, who did a lot of this work. And what you see here is a heat map of the genes that were expressed in the blood, and these were all immune genes. So we did a panel of about 500 of these genes, and on the left-hand side you can see that the healthy controls, those were the genes that were turned on in red, and those were the genes that were turned off in blue. And you can straight away see that in case 1, 2, and 3, the patterns were quite different from healthy controls. And remarkably, case 1, especially in this part, was a lot more red than in cases 2 and 3, suggesting that maybe genes that clustered into the toll-like receptor or inflammatory signaling pathways were overexpressed in case 1 compared to 2 and 3. And that's very consistent with a lot of the literature that suggested that COVID-19 is really an inflammatory response to SARS-CoV-2 infection. And maybe that's what is driving disease, and if we can inhibit the inflammatory signaling, then we should be fine. But actually, upon closer look at this, you can see that actually the worst day of disease in case 1 was in day 5, right? It's indicated by the arrow right at the bottom. Whereas the day with the highest expressed inflammatory genes was on day 6. So actually some of the genes actually happened after the event, right? So not all genes led up to the worst day of disease. So if you now look back at what happened at day 4, that could potentially lead to what manifests clinically in day 5, then the only thing that we could find quite consistently throughout the pathway was the interleukin-1 response. And IL-1, we know, is a pro-inflammatory response cytokine. And so both IL-1 alpha and beta in left and middle of the graphs, you can see that the blue line, which is case 1, was higher above the controls, which is the bar in gray right in the middle, as well as cases 2 and 3 at the bottom in red and yellow. And what you can see is that at day 4, these IL-1 was already much higher than control levels, and that peaked at day 5 and it came down. IL-1 beta was highest in day 4, and then certainly above the control levels, and likewise the receptor. So all that suggests that maybe this is, it's not all inflammatory genes, but a specific pathway that could be driving disease. So then the next question is, what drives these pathways? And we know that COVID-19 or SARS-CoV-2 infection isn't the only infection that causes this spectrum and disease outcome. And I work mostly on dengue. I'm going to use dengue as an example. We know that when amongst say 100 people infected with dengue, only about 1/3 will manifest symptoms, and the remaining 2/3 will have no symptoms whatsoever, will not even realize they're sick, or what we call asymptomatic. Right? And so the symptomatic group is actually the very tip of the iceberg, and certainly the severe disease is then right at the very top, whereas most infections actually do not cause any symptoms. So then what differentiates these people? So we, of course, have been interested in trying to understand how the virus interacts with the host cell. Now, if you think about it, viruses are the classical parasite, right? Because outside of the cell, they are not alive. They can't do anything on their own. They need to come inside the cell and then hijack the cell's machinery and then replicate itself and then come out, and that's how it perpetuates itself. And once inside the cell, it makes a lot of these interactions inside the cell so that one, it replicates itself. Two, it suppresses all these host response that would otherwise kill it, right? And it's all the interactions that make it a successful virus to infect humans. So then we're interested, of course, in what aspects of these pathways underpin why some people get sick and why some people do not. But when you study COVID-19, you study dengue, when you enroll the patients, 5-6 days would have gone by since the time they were infected until the time they develop symptoms. And so once they develop symptoms, all these processes, of course, are already in place. All the pro-inflammatory genes, like the heat map I showed you earlier on, are already in play. What led up to that? Well, the only way you can understand that is to catch them right at the point when they got the infection. But that's very hard to do with natural infections. So to understand that, we've been using the yellow fever vaccine to study this. The yellow fever vaccine is a live vaccine. So it actually is a live virus that will cause a very mild infection that in most people will not make you sick at all. Right? And in some individuals, about half of them will develop a 1 day worth of side effects. And these side effects is like this flu-like illness is what I'm going to show you here. Then most people, in the colored in red here, will report 1 day worth of feeling a bit tired, maybe a bit of headache, some bit of joint pains, and a bit of fever, which will resolve within 24 hours, and then they're fine. What this graph shows is when the symptoms appear. So this middle line here is the median time on which when the people who receive the yellow fever vaccine will report these symptoms. And the median time is about day 6. Okay, so 6 days after you get the yellow fever vaccine virus, which causes an infection, 6 days later, you get symptoms. So very similar to what dengue is like, right? About 5 days, COVID about 5 days. So it takes about 5 days for the virus to interact with the cells before the symptoms then appear. Okay, so then this gives us a model to understand why some people get sick and some people don't, or some people get symptoms and some people do not. So right off the start, we do not find an association between the amount of the virus in the blood versus those who get symptoms or those who do not, right? So you can see on the graph here, day 3, day 7, there's no significant difference in the levels of the virus in the blood as measured by RT-PCR between those who develop symptoms in red and those who do not have symptoms in blue. What differentiates those who go on to develop symptoms or not at day 6 is the response to the infection at day 1. Right? So this is the point of time in which they develop these symptoms. 1 day after you give the virus, yellow fever 17D, then the host response at this time, shown over in this heat map over here, determines whether you're going to get the symptoms or not. So what you see here, AE means those who develop symptoms, and blue are those who have no symptoms. And straight away, just right off the bat, if you look at this heat map, more of those in the symptom sites are red, and more of those in the without symptoms are blue, and red means the genes are switched on, and blue means they're not. So right off the bat, this trend just suggests that, and if you now put it into these pathways, would suggest that very early, within 24 hours, the activation of the innate immune response then are correlated with those who then go on to develop symptoms. Okay, suggesting that that's the process. Okay, then why is it that not everyone develop this innate immune response? Only some do and some do not. but everyone got the same virus and everyone got about the same amount of virus. So why only some? So obviously, therefore, there must be something at baseline. Before they're even infected with the virus, there's something going on that then upon virus infection triggers this process very quickly within 24 hours. Because this is the innate immune response, the obvious thing to do then is to ask, is the innate immune response activated in these individuals who go on to get symptoms versus those who do not? And the answer is no. None of the innate immune genes, when you measure it in the blood, were significantly elevated in a symptomatic group compared to the asymptomatic group. So if you now take a step back and say, okay, it's not the innate immune response, let's look at all the genes that we can measure in the blood. And then ask which ones were higher in those with, which ones were higher or lower in those who have symptoms versus those who do not. And the answer is in this slide, right? So on the left-hand side are those genes that are upregulated, in other words, more expressed in those who develop symptoms, who go on to develop symptoms. And on the right-hand side are those genes that were lower in those who go on to get symptoms. And this is before you give the virus. And so if you look at these pathways at the top, then what you see is the unfolded protein response or chaperone, everything to do with ER stress or endoplasmic reticulum stress. And I'll explain what that is in a minute. And this thing called submyelination is related. Okay? So what ER stress and submyelination and all that are supposed to be doing is that your endoplasmic reticulum or your ER, you can think of it like a factory. And that's where the proteins are made. Right? And like any factory, you have to have a quality control because you want to know that the product that comes out of this factory is good. Right? Now, when the products are faulty, then what you do is you go back and you slow down the production, figure out which part of the process is faulty, remove all the faulty products, make sure they're not exported out, and then you make sure that all the quality controls are in place to ensure that what comes out is good. And that's exactly ER stress. So it triggers a response and it slows everything down, it tries to get rid of all those faulty products so that it makes sure that everything that then comes out is corrected. On the right-hand side, if you look at the top pathways, you have all these lipid metabolism as well as Krebs cycle or citric acid cycle or tricarboxylic acid cycle activity, which is in the mitochondria. Right? So on the one hand, it seems like your cells are more stressed. On the other hand, energy levels or these production of energy using through this TCA cycle is now lower in these individuals that go on to get symptoms. So that was done in a clinical trial settings. These are all humans. These are not animals. So and they were healthy at baseline, right? And so we did that in a clinical trial to make sure that was not unique to one cohort. We did it again in a separate trial. So what you see is trial 1, trial 2, and the results are reproducible across the two trials, right? Again, the ER stress as indicated by UPR and submyelination. And the second one, we measured specific genes in this ER stress pathway. Again, they are significantly higher in the red, which are those who went on to get symptoms, versus the blue. Likewise, metabolism, except that the lipids now, if you take all the genes in these pathways, then there's no significant difference in the symptomatic versus asymptomatic group. But the TCA cycle is still lower in those who went on to get symptoms, right? And across two trials. So then what's happening? In fact, the difference in the expression of these ER stress genes in the ER stress pathways as well as the TCA cycle is so different that you could actually predict the outcome, right? So what you see here is the ROC analysis, and these are the area under the curve values, so it's about 0.7. They are all statistically significant. The way to look at AUC is that if it's an inverted L, it's a perfect test for a condition. And this red line here, red dotted line, is where there's absolutely no difference, right? And so the more it skews towards this perfect L, the better the test. So what you see here across all these three genes in these three different pathways is that they're not perfect, but they do predict the outcome to a reasonable extent. Right? So this is before you even got the virus. So the next question is, these are all genes. Can we measure actual substances that's as a consequence of the differences in these gene expression of these genes. And so because the metabolism was involved, we then did a metabolome analysis. So we measured all the metabolites in the blood. And again, what you see here on the left-hand side is, the different groups, right? So in purple and in light blue here, which is right at the bottom, are those who go on to develop symptoms versus those who did not get symptoms. And this before they got the yellow fever vaccine infection, and this is 1 day after. Again, you see that the segregation is almost perfect, right? There's almost no overlap in these groups, that you can tell those who go on to develop symptoms versus those who did not before you even give them the vaccine. And if you ask, okay, amongst all these metabolites, which ones is the most significant? And it turns out that it's the TCA cycle. So the TCA cycle, or Krebs cycle as we know it, is a way in which we process our glucose, our sugar, and then all the way to making energy, right? And so the TCA cycle is represented over here. The glucose is metabolized, the pyruvate, and then eventually to form acetyl-CoA, which is then all these processes are then happens in the mitochondria. And so if you look at the key metabolites in this pathway, citrate, isocitrate, and malate, they are all lower in those who go on to develop symptoms versus those who did not. These were all measured by mass spec, so we also measured them in a separate method just to make sure that it's not unique to one method, but in a separate method you can still see a significant difference in the citrate levels. And that because the starting point is glucose, we also measured glucose in the blood, and there was no difference in the glucose level, suggesting that for some reason the TCA cycle activity in these individuals are lower in those who go on to develop symptoms from infection. So to summarize everything that I've talked about so far, if you give the yellow fever 17D vaccine to say 100 people, about half of them will develop symptoms, and these 50% develop symptoms. They do so because 1 day after vaccination, they trigger an innate immune response. And the reason why they do so is because they have, at baseline, before you even give them the yellow fever 17D vaccine, they already have some level of ER stress, and it says adaptive because they can adapt to it. It's not overwhelming, so that they can manage it. And part of the adaptation may be a reduced TCA cycle activity. Okay, so then how does this lead to this? So what happens after you give, you get infected with a virus is obvious, right? Because the virus now needs to replicate. To replicate, it needs to make new copies of itself, and what the protein shell is a critical part, as well as the enzymes to replicate the genome. And so the first thing, that the top pathway in 24 hours after you give the virus, if you look at all the genes that are turned on in the body, then translation is top, right? So it makes new proteins. But also TCA cycle, it needs energy to replicate the virus. So everything else, the rest are all down. But these three pathways are exactly what is related to what happens at baseline. So in other words, if we put all that information together, then if in those individuals at baseline before infection, they already have some level of ER stress, they compensate for the ER stress by reducing their energy production through the mitochondrial activity. Then upon infection, which increases translation and therefore protein production, and because the virus needs energy, so it's going to turn all these energy pathways on, it's going to increase ER stress further, and it's going to increase the energy demand. Now the cells cannot compensate. So it goes into this thing called the maladaptive ER stress. It just kills itself. So something is in here so wrong, the quality control cannot be managed, it will just shut down and it kills itself. It undergoes this programmed cell death. And so the key pathway in which it does this is through the reactive oxygen species, or ROS response. So if you come back to our cohort of volunteers, if you look at in blue, those who did not develop symptoms versus those who did in red, then this is a way of measuring for ROS response. It's a byproduct of it called the methionine sulfoxide, which is present in the blood. And so before vaccination and after vaccination or after infection, you see a significant increase in methionine sulfoxide levels compared to those who develop no symptoms, which is there's no difference in this cohort. Another way of measuring ROS is to measure again a byproduct, superoxide dismutase. Again, you don't see a difference in those without symptoms, but you do see a significant difference in those who go on to develop symptoms. Then finally, at the genetic level, how our cells control the ROS response is to express a protein called thyroredoxin. This suppresses the ROS response. So if you want to turn on the ROS response, you have to suppress this thyroredoxin, and it does this through this protein called TXNIP, or thyroredoxin interacting protein. This then binds to this, and then it stops this from functioning, and so then the ROS is triggered. So if you then ask at the genomic level whether TXNIP is increased in expression in those with symptoms and those without, 1 day after infection, then the answer is yes. So across both trials, you can see that the red dots, which are those who develop symptoms, are much higher than those who do not develop symptoms. In fact, if you then ask at baseline the ER stress genes, if you take all of it and give it a score, it is correlated with the TXNIP level 1 day after they are infected, and this applies to both the ER stress as well as the stimulation genes, right? So if I put all this together and I'll summarize this, what happens that at baseline in those individuals with ER stress, then when you're infected with a virus, then that virus is going to need to make more copies of itself. To do that, it needs to increase the translation activity in the endoplasmic reticulum, as well as it needs energy. And in those who already have some level of ER stress and they are trying to adapt to that, then this additional demand on ER as well as energy then tips the cells over into maladaptive ER stress, triggers an ROS response. The ROS then triggers the innate immune response, and that then eventually cascades to symptoms. So finally, I just want to come back to this point. And okay, so then why is it that some people have increased ER stress and lowered TCA cycle activity before they're even infected? So what drives that? So we don't know the true answer yet, but I think there are several factors. First of all, obviously genetics. Maybe some of us are more genetically susceptible to these states. BMI, and I'll show you some data, preliminary data on that. Age, perhaps, because we know that in aging, ER stress is increased and energy levels go down, and maybe that's all associated with this. And maybe that's why in COVID-19 patients, the older you are, the worse the disease. Possibly the microbiome. We don't really know how the microbiome affects or interacts with the immune system in general, but we know it does. But the mechanisms and all that are still unclear at this stage. Perhaps also others that are not listed here. So just to give you an idea, we love to explore the impact of age on ER stress and TCA cycle activity and how it then affects infection outcome, but we have not been able to do that. What we have done is go back to our data and look at BMI. Right? And so we do see an association between BMI, as indicated in the x-axis, and a couple of genes in the ER stress pathways, and these are all positively correlated. So the higher the BMI, the more ER stress genes are expressed. And then we also found that if the BMI is higher, or at least amongst the symptomatic subjects after they receive the yellow fever vaccine, they have an average a higher BMI than those who did not develop symptoms. In fact, if you stratify our cohort by BMI, you can see that the proportion that develop symptoms after being given the yellow fever vaccine is increased with increasing BMI. Right? So this is unpublished data, and we hope to be able to follow this up with additional studies and understand how BMI affects ER stress and then eventually symptomatic outcome. And certainly, obesity has been suggested to be a risk factor for COVID-19 as well. So coming back then to this summary, we know that this is that there's a baseline susceptibility factor, the symptomatic outcome, right? And it's partly driven by this innate or immune response or the inflammatory response that eventually leads to symptoms. So if given what I've said, then if you think about it, then really the way to treat disease is to really target this host response, right? But actually, if you think the real trigger, I mean, although these are all susceptibility factors, the trigger is still the virus. So the question is, if you treat the virus, will you change these outcomes? Or do you really have to go and target the innate immune response? Or do you have to target ER stress? Well, the answer is you can still target the virus. And we finally managed to kind of put all of this together because we had a new candidate therapy for yellow fever. And this was in a yellow fever antibody that we, and I say we because Tycan generated. And this went to phase 1 trial. And because yellow fever vaccine causes an infection, we could use that vaccine as a way of simulating an infection rather than test this in yellow fever patients. So this was published just a few weeks ago in New England Journal of Medicine. And the data is here. So the red line is placebo, blue line is those treated with this antibody called TY014. The x-axis is the proportion without symptoms. So you start off at 100% of people without symptoms, then they're infected. 1 day later, we treated them with TY014. Those who received the drugs continued to be asymptomatic, whereas those who got placebo, then they started developing symptoms, and 80% of them will go on to develop symptoms. Viremia, not symptoms, sorry. The PCR data is over here. Again, you can see that in placebo, 80% of them develop detectable, almost 100% of them develop detectable RNA. Up here is 80% develop infectious viremia. Whereas those who are treated had absolutely no detectable RNA or infectious virus. So the previous slide, I showed you that TY014 completely knocked down the virus, right? And this is 24 hours after. So in other words, if you put what I've said earlier, that all that process would have been triggered, the ROS, everything would have been triggered, right? And then you come in and knock the virus out. So the question is, can you still change the outcome? And the answer is yes. So if you knock the virus down, as I showed you in the previous slide, then over in this slide, you can see that those who got placebo, all the genes continue to be turned on, whereas if you remove the virus, they then get switched off. And correspondingly, the proportion who develop symptoms continue to increase with placebo, whereas if you stop all these events, then the symptoms disappear. So I think at the end of the day, the outcome of infection is there are clear susceptibility factors, and I think we're just beginning to scratch the surface. So ER stress and metabolism being two related factors. Then on top of that, when you have an infection, it triggers the increased protein production and energy demand. Then it tips the whole cell over, but that would suggest that maybe we need to dampen the innate immune response to prevent disease. But at the same time, you can still get rid of the virus. And the earlier you can get rid of the virus, the more of these processes you can stop, and we can make a difference to the disease outcome. So finally, I just want to acknowledge all the people who have really contributed to this work, because without them, I would not have been able to tell you this story. So, finally, I just want to thank Merck for the opportunity to present this work, and all of you for sitting through this talk. Thank you very much.
Thank you, Dr. Eong, for a fantastic presentation. What a way to kick off this session.